So our keynote speaker for this afternoon is Dr. Gabriella Gobbi, who is a professor in the Department of Psychiatry at McGill University, where she holds a Canada Research Chair in Therapeutics for Mental Health. She leads a laboratory in basic neuropsychopharmacology while working as a psychiatrist at the McGill University Health Center. Her research spans from bench to bedside, bridging the gaps between fundamental and clinical research. She has won several prizes in the field of neuropsychopharmacology, including the 2022 CINP Sumitomo Synovian Brain Health Basic Research Award and the CCNP Innovation in Neuropsychopharmacology. Dr. Gobbi's work explores neurobiological mechanisms implicated in psychiatric disorders and how pharmacological interventions, including psychedelics, the focus of the talk today, may target those mechanisms to treat mental health disorders. I'm sure you'll find her talk very engaging and exciting. I'll now pass on the floor to you, Dr. Gobbi, for your keynote presentation.
Okay, thank you, everybody. Thank you, Tony, Marisha, Lily, and the team for this invitation and for this great organization. It is a pleasure today. I'm here on Phoenix. I'm participating now at the ECMP, but I'm here now with you to talk about psychedelics, the promises and pitfall in translational research. So let's start with my conflict of interest. My research on LSD is sponsored by mostly CHR, FRQS, and Alan Edward Foundation. But recently I received also a grant from Diamond Therapeutics for studying psilocybin. Let's start with what is psychedelics. Psychedelics are also called classical hallucinogenic drugs and are mostly 5-HT2A receptor agonists. We have also NMDA or ecstasy that is considered a class apart. In the last year, as you know, we have several RCT study published with the psychedelics. Most of them have demonstrated that psychedelics can be a new avenue for the treatment of mental disease. The most known is the MDMA RCT study in PTSD, and the study has demonstrated that MDMA can treat severe post-traumatic stress disorder. Another study was published recently about alcohol use disorder with psilocybin. Another one was published 1 year ago and demonstrated that psilocybin versus escitalopram also improve major depression. Another study also was published in August, demonstrated that LSD can be a good medication for anxiety with or without life-threatening illness. Psychedelics are a very important topic and also represent a novel paradigm in therapeutics. Why? Because in low doses, LSD, psilocybin act like a psychotropic drug by binding 5-HT2A, 5-HT1A, 6 receptor, and play on neuroplasticity, epigenetics. But at another level, these psychedelic drugs can also act by modulating the brain circuits, in particular the thalamocortical circuits and the DMN. It has been hypothesized that because psychedelics activate these circuits, it is possible to work better in psychotherapy. There is a better appraisal, a better integration when a patient is under psychedelics, because there is a decrease of the activation of DMN and an increased activation of cortical thalamic circuits. Then I will talk a little bit more later about this. But because of this complex mechanism, the psychedelics can really represent a new paradigm in mental health. So the objective today will be to understand the effect of LSD in social behavior, in anxiety, to understand the mechanism of action of this effect, and to understand how the LSD can modulate brain circuits and mind. As you know, the LSD was first synthesized in 1938 by Albert Hofmann. He was studying a new drug for the cardiovascular system by the ergot fungus, but finally he synthesized this novel drug. He tried himself the LSD, and then also he wrote a book called My Problem Child. And he also described the effect of LSD as plants of God. He used to call LSD as a god because it was also inspiring him some mystical thinking and spiritual thinking. We know from the studies of the 60s that LSD can increase empathy, social behavior, and in the 60s was also used for treating autism disorder. In my laboratory, we ask in preclinical model, in animal model, if LSD can modulate social behavior and anxiety. The first problem that we had in our lab was which dose of LSD we can use in mice, because we didn't have so many studies say which dose should be used. So we decided to perform some experiment with electrophysiology, studying the serotonin neurons that are localized in the Raphe nucleus and the dopaminergic neurons of the VTA. So using electrophysiology, we found that low doses of LSD by 5 to 20 microgram kilo were able to modulate the serotonin neurons, but not the dopaminergic neurons. You can see here you have a dose response with serotonin, but no effect in dopamine. However, at higher doses, LSD was able also to modulate the dopamine. However, we decide to stay with the low doses because we want to use to exploit the effect of LSD on serotonin system. So we use low doses, and we see that low doses in acute decrease serotonin firing activity, but after 7 days of repeated treatment, there was an increase in serotonin firing activity, the same increase that we notice also with antidepressants, SSRI, that are used to treat depression or anxiety. So we tested these doses of LSD in social behavior. We performed the direct social interaction test that consists in measuring the time of interaction between two mice, and we observed that after LSD treatment, the social interaction was increased, but not after acute injection. We then perform another test that also assesses the social behavior. Is called a three-chamber sociability test. The mouse first is exposed to a stranger one, and in this case we measure the sociability, and later the mouse is exposed a new stranger, and in this case is assessed the social novelty, the curiosity for a new stranger. We observe that LSD not only increase the sociability index, but also increase the social novelty. So until now, we can say that low doses of LSD at 30 microgram kilo for 7 days increase social interaction, increase the preference for a social stimulus, increase preference for social novelty, and increase the serotonin firing activity. Then we ask what is the neural basis of these changes? And we decided to study the medial prefrontal cortex. We know that the medial prefrontal cortex is an important area in the social cognition, is rich in serotonin 2A receptor, and has several implications also in autism. So, first of all, with the microiontophoresis, we tested the responsiveness of some receptor after 7 days of LSD. And we noticed that after 7 days, the responses of 5-HT2A as well to the AMPA are increased, but not the response to 5-HT1A receptor and NMDA. We did also some experiment using the optogenetic. We inhibited the prefrontal cortex, the glutamatergic neurons of the prefrontal cortex, and we noticed that after the inhibition of these neurons, LSD no longer worked. However, when the photoinhibition was put off, we can see that LSD increased the social behavior, and also the vehicle group come back to the baseline. So this experiment demonstrated that the prefrontal cortex is very important, not only for the basal social behavior, but also for the prosocial effect of LSD. Since in our previous experiment, we also noticed that LSD increase 2A and AMPA responses, and we know that AMPA responses is linked to the activation of the mTOR complex, we decide also to study the mTOR complex. mTOR is also important for the mechanism of action of other fast antidepressant drugs, as the ketamine, for example. So we decide to measure the mTOR, and we find that after 7 days of LSD, not only the mTOR was increased, but also the PAKT that is linked to the mTOR complex pathway. Then we use these knockout mice called raptor mice. Raptor is part of the mTOR complex. And in the mice without the mTOR at the level of glutamatergic neuron of the prefrontal cortex, the LSD no longer worked. So this experiment means that mTOR in the prefrontal cortex in glutamatergic neurons is very important for the action of LSD. Not only this, but in this raptor knockout, the LSD also was not able to increase the response to 5-HT2A receptor and AMPA receptor. So we can say that for the prosocial effect, LSD works through the 5-HT2A receptor that also activate AMPA and activate the mTOR in glutamatergic neurons of the prefrontal cortex. Next, we also explore LSD in anxiety. We know that there are 2 papers that have demonstrated that LSD can be an anxiolytic in patients with anxiety, life-threatening disease. So we did this experiment in mice. Some mice underwent a chronic stress procedure for 15 days, restraint stress, and we administered LSD from day 7 to day 15. So what we found that LSD improve anxiety induced by stress. Here you can see that after treatment with the LSD, the time in the center is increased, as well as the entry in the center in the open field test. We perform also another test to measure anxiety, that is a suppressing feeding test. And also in this test, LSD was able to prevent the anxiety induced by stress. Then we ask if LSD can work as SSRI. As I mentioned at the beginning, SSRI in acute decrease the serotonin because the 5-HT1A receptor are activate, but after long-term treatment with the SSRI, the 5-HT1A receptor are desensitized, and the serotonin increase. So we found that LSD did similar things. After acute injection, decreased serotonin, but after 7 days, the serotonin is increased. Not only, also the 5-HT1A autoreceptor, as for SSRI, are desensitized. Not only this, but in case of stress, the serotonin is decreased, and the LSD was able also to reverse the decrease of serotonin caused by stress. So, since a lot of antidepressants also act in spinogenesis and neuroplasticity, we decided also to measure the spines in stressed animal. And we found that not only LSD increases spines in control animal, but more important, increase the number of spines that were lost during the stress. After this, we decided also to test if LSD also change some epigenetic, the regulation of epigenetic, and this regimen of LSD hypermethylate or hypomethylated some genes. And the genes that were methylated, very interesting, were genes linked to the neurodevelopment of the central system, formation of the brain, cell viability of neurons. In parallel, we did also a study of proteomics, very interesting. The protein that were upregulated or downregulated were protein linked to the synaptic plasticity, to the regulation of receptor-mediated endocytosis, and very interested to the neurotransmitter receptor internalization and regulation of neurotransmitters. Then we studied in particular 4 proteins that were mostly changed in the epigenetic studies. And very interesting, these 4 proteins that are protein always linked to the neuroplasticity, we found also an increase of mRNA level and increase of protein level. It means that these proteins were really expressed, it was functional after LSD treatment. So here my conclusion of this first part, we can see that repeated LSD administration enhances sociability. This is mostly acted through the prefrontal cortex, and mTOR complex is also involved in this effect. LSD has anti-anxiety properties similar to the SSRI. They restore the serotonin firing activity after stress. And LSD has a neuroplastic and epigenetic also properties. Now I talk a little bit about the second level of psychedelics, that is the level in brain circuits. So as I say before, the modulation of circuits is another important component of a psychedelics mechanism of action. One of the circuits that were more studied was the DMN and the thalamocortical circuits that are both implicated in consciousness. The DMN, this default mode network, and the CSTC, that the cortical striatothalamo-cortical loop, or CTC. So for people that are not familiar, the DMN is a network that is very active when our mind wandering, when we walk in a park of just thinking about nothing. And the DMN, when we are in this wandering state, is very, very active. It is also implicating in many functions of the self, as the autobiographical information, self-reference, emotion of oneself, in thinking about others, also remember the past and the future experience. So it's a system that is really linked to the self, to the perception of the self. Very interesting, it seems that under psychedelics, the activity of DMN collapse, decrease. And the people that is expert in psychedelic assisted psychotherapy think that this decrease in the DMN attention, in DMN activity, is the background that allow psychotherapists to work in the novel perception of self and past experience during the psychotherapy. Another interesting circuit that is implicated in the mechanism of action of psychedelics is the thalamus. Also, my laboratory was very curious about the thalamus, because the thalamus is the part of the brain that connects the external stimuli with the internal integration. Is also involved in the consciousness. For example, during the deep sleep, the reticular thalamus is very active and send some input to the cortex, and the cortex is shut down. Also in the state of coma, the thalamus is very compromised. Very interesting brain imaging published by Mueller in 2017. They found that under LSD, the most involved circuit is exactly the thalamus-cortex loop. It seems to be hyperactivated under LSD. In mice, in my laboratory, we found a little bit a similar curious things. We found that some neurons that high firing are decreased by LSD, while neuron low firing are increased. The same things happen also for the burst activity of these neurons. But this effect, very interesting, was only observed at high doses, as if the mind altering property of LSD probably occur only at high doses. The same things, similar things we found also with the psilocybin. So now, just to summarize, it's very important also in this nice review by Vollenweider and Preller. It seems that the circuits, in particular the circuits of the thalamus, is also implicated in the mechanism of action of psychedelics. And psychedelics, as I mentioned at the beginning, works not only through this pharmacological modulation of serotonin receptor, dopamine receptor, but also through this mind-altering property by allowing decrease self-order differentiation, decrease in rumination, for example, during the depression, by increasing empathy, by increasing also a novel reappraisal of the past experience, negative experience. Finally, this is my last question. Everybody asks if the spiritual experience is very important for psychedelics. I think yes. I think psychedelics, a spiritual experience, yes, must not be seen as a belief or not believe in God, but as the highest expression of the human mind. So spirituality is when the brain think about unity, insightfulness, blissful state. And as it was demonstrated, this effect is mediated by the 5-HT2A receptor. And in the recent clinical trial, citalopram, psilocybin, the patients that were more responder were the patients that also have this spiritual experience. So what next? Of course, we have to translate all this data in preclinical data in clinical data. We need to do more clinical trial to see if LSD can be good for social anxiety, for generalized anxiety, also to understand better how LSD can really modify these circuits. And the big question also today, I hear the ACMP, everybody asks if we really need psychotherapy to exploit the therapeutic use of psychedelics, so maybe it's possible just to use alone in low doses. This is a big translational question that we have to answer in the next days and next year. Finally, thank you to my team, in particular to Danilo De Gregorio, Michael Pileggi, and Antonio Inserra that did the most part of these experiments, all the team. And finally, please don't forget that we have the CIMP in Montreal next year from the 7th to 10th of May. You are welcome. We have a lot of prize fellowship for students and for young investigator. Please come to Montreal next year. Thank you.
Thank you so much, Dr. Gobbi. It was really interesting that you brought up the question of like the spiritual experience, because that was very heavily debated, and now we're seeing evidence that the mystical experience does kind of predict therapeutic efficacy. So thank you so much for bringing that up and for your talk. So we do have about 8 minutes for questions. If you do have questions, you're welcome to put them on Huba or into the Zoom chat. We do have 2 questions already on Huba, both from Lily Alexandrova, who you know. She says, you mentioned some in your presentation, but what are some animal models or behavioral assays that have been useful in studying the effects of psychedelics? How can we improve preclinical models and studies to make them more clinically relevant?
Mm-hmm. So if I well understood, she asked how our preclinical model can be more translational for the clinical. Is correct?
Yeah, like how do some preclinical animal models, how could they potentially be improved, or how well are they working now to ensure that our preclinical studies are more clinically relevant?
Okay, so it's not so difficult because
a feeling of warmth and closeness to people when you do any of those things? No. Okay.
Okay. Okay. So we can use for animal model all the animal model that we already know in psychopharmacology. At the end of the day, here in our experiment, we use the animal model for anxiety, or not for anxiety, as the open field, the novelty suppressing feeding. You can use the model for depression, the model for social interaction. We use exactly the same model that we use for all the psychotropic drug in psychopharmacology. The big question with the psychedelics is, for us, is the dose. It's not easy to translate the dose from animal to human, from human to animals, because rodents are not the best model for 5-HT2A because of less 5-HT2A than humans. And we have to use much higher dose than humans because rodents metabolize very rapidly these compounds. This is a problem. The second problem is the problem how to assess the hallucinogenic properties. While for 5-HT2A agonists as LSD and psilocybin, we can use the twitch to assess the hallucinogenic potential. This is not valid, for example, for ketamine or for an NMDA. So for this other compound, we have to select other kind of tests to evaluate the potential dissociative state. As for example, we can use the locomotion, we can use the PPI. The PPI is a very valid model to assess the potential dissociative state. So we have to use, let's say, in a more complex, creative way, all the test paradigm we know already for psychopharmacology.
Thank you so much. One more question on Huba is, this is again from Lily, as a multidisciplinary researcher that works at the interface of preclinical and clinical research, what are some major practical barriers to translational research along the bench to bedside to community drug development pipeline? What are special obstacles when a drug is a controlled substance?
What are the obstacles?
Yeah.
Okay, the obstacle are, I can say the same obstacle that we have in general in translational medicine. The main obstacle are not is not science, is mostly the interaction between people that do science. We need more interaction between basic researcher and clinician. When we have together these people together, a lot of novel ideas can come. However, I must say for psychedelics, there is, yes, a big limitation because, for example, the spiritual mind, we can study the spiritual mind in mice, in rodents. Rodents has not the sense what is the meaning, what is the blissful state, etc. This, of course, is a big limitation of the study of psychedelics.
Thank you so much. Yeah, one thing that the MATRIX-N cluster is trying to do is build those connections and interactions between preclinical and clinical researchers and clinicians as well, to help bridge some of those gaps and develop new collaborations and ideas.
Yeah.
I think we have time for one more question from the audience, if anybody would like to put it in the chat or speak up. All right, I have one quick question, and then the next session does start at 2:20. So if you need to leave to go to that, you can go ahead, everybody. But how do you find that your clinical work, because you're working as a psychiatrist, how do you find that your clinical work influences your research and vice versa?
My clinical work influences my research. Okay. Yes, I decided quite early in my career during my medical school that I want to be also a researcher, because just to be a clinician was not enough for me, because especially in psychiatry, you have always a lot of questions. You want always to do more for your patients, for people for which there are no real final treatment, as a schizophrenia, as autism, etc. So I decided to go also in research to bring more to my patients. And then at the same time, my experience as a researcher, even if with animal, with basic research, actually brings me a lot of knowledge that I can really transfer to my patients, to find a new solution to my patients. And at the same time, my patients bring a lot of questions that I can answer in research. So it's very important, especially for the younger generation, for the new student, that you have in mind also this possibility of a career, to combine both clinical research and basic research, because this is the real translational science. When a person is able to pass from a hat to another hat and bringing together different knowledge.
Yeah, I really couldn't agree more. Anyway, it is 2:20. So Dr. Gobbi, again, I just want to say thank you so much for presenting, especially it sounds like you're maybe at another conference today. So I'm wearing two hats, or in two places at once today. So thank you so much. And everybody, you are welcome to head off to your next session, whatever that may be. Thank you so much.
Thank you. Bye. Bye-bye.